It's like when you see a city from way up high,
Mm.
like from an airplane window,
Uh-huh.
it's impressive, right? All the streets and buildings, but you
Yeah.
just know you're missing all the good stuff, the nitty-gritty down on the ground.
Right, right.
And that's what's so cool about this research we're diving into today.
Yeah, this is
It's like we're finally zooming in from that airplane view of cancer
yeah.
and getting down to street level. Like we can see the individual cancer cells now.
It really is a whole new perspective. We've been stuck with the big picture, the broad strokes for so long.
Right.
This paper though, they're using single cell RNA sequencing and
Oh, wow.
it's like ditching those clunky old magnifying glasses and finally getting a high-powered microscope.
And what do we see with our brand new microscope?
Things we could never see before, that's for sure.
Haha, I bet. Okay, so this paper focuses on what are called cultured cell lines, which I guess are kind of like the lab rats of cancer research, right?
Exactly. Standardized cells, easy to grow in the lab, so scientists use them for all sorts of experiments.
So they're like the control group, the baseline.
Yeah, you could say that. And in this study, they looked at almost 200 different cell lines covering 22 different types of cancer. I mean, talk about a deep dive.
Wow, 22 different types. That's wild.
Yeah.
But I'm guessing they found that even these lab-grown cells, even in these controlled environments, they're not all perfectly identical clones.
You're catching on quickly. That's exactly what makes this research so fascinating. They discovered that even within these cell lines, which are supposed to be uniform, there's this surprising amount of diversity.
Interesting. So even if you have, say, like breast cancer, not all the breast cancer cells are exactly the same. So what? Why does it matter so much that there's all this, what do they call it?
Intratumoral heterogeneity.
Right, that. Why does that matter so much?
Well, just imagine trying to fight an enemy that's constantly changing its tactics. Like every time you think you have them figured out, they switch things up.
Oh, that's a good analogy.
That's cancer treatment in a nutshell. And this diversity, this heterogeneity, it's a major reason why some treatments work for some patients and not others, and why cancer can become resistant to therapies, even ones that were working before.
It's like it's always one step ahead. So knowing that there are these different, like neighborhoods within a tumor, each with its own character, could that be the key to developing better treatments, more personalized ones?
Now you're getting it. Absolutely. This paper takes a big step in that direction. They identified two main types of diversity in these cell lines: distinct subpopulations, which are like, as you said, distinct neighborhoods, each with a unique set of traits.
Okay, following so far. Distinct neighborhoods. So that's one type. What's the other one?
The other is what they call continuous programs of heterogeneity, or RHPs for
Yeah.
short, because nobody wants to keep saying that whole mouthful.
RHPs.
Yeah.
So those are like the other type of diversity they found.
That.
But those sound kind of complicated.
They are a little bit, yeah.
So what are they exactly?
Think of it this way. Instead of hard boundaries between one type of cell and another, it's more like a spectrum of behavior. Cells can transition between different states along the spectrum, you see, changing their behavior and function in response to their environment, or maybe signals from other cells.
So it's not just that there are different types of cancer cells, it's that they can like change and adapt on the fly, almost like they're evolving right there in the tumor.
Precisely. That's a great way to put it. And here's what's really wild. The researchers, they identified 12 of these RHPs in the cell lines.
12, wow. Okay, so 12 of these programs, these spectrums of behavior, and what do those programs actually do? Like, what are they about?
Well, that's what's so intriguing. 9 of the 12 RHPs that they found in cell lines, they also popped up in real human tumors.
Whoa, really?
Mm.
So it's not just a lab thing. These RHPs, they're not just some weird quirk of growing cells in a dish.
Right, it's the real deal. This is giving us real insight into the nitty-gritty of what's happening inside tumors in real patients.
So we're not just looking at a bunch of random chaos. These programs, these patterns, they're repeating, which means they probably matter a lot.
Yeah.
They could even hold the key to how cancer survives or maybe even spreads. Okay, I'm hooked. So what were some of the most interesting RHPs they found? Give us the highlights.
Well, one that really stood out, they called it the stress response program.
Stress response program. Okay, let me guess. Does that mean what I think it means? Like, are some cancer cells actually getting stronger because of stress? Tell me I'm right.
You're absolutely right. They found that some cancer cells actually get turned on by stress, things like damage to their DNA.
Wait, what? That doesn't make sense. Shouldn't that kind of damage kill a cell?
You'd think so, right? But these cells, they thrive on it. It's like trying to fight an enemy that gets tougher the harder you hit them.
So blasting all the cells with treatment might not be the best approach then.
Not necessarily. Not if you're dealing with these stress-loving cells.
This is where our whole city from above analogy really falls apart, huh?
Yeah.
If we only look at cancer from a distance, we miss all those crucial details. So we've got the stress response program. What other kind of craziness is going on down at the cellular level? What else did they find?
Well, there was another group of RHPs, kind of a fascinating trio. They dubbed them the EMT trio.
The EMT trio. Okay,
Yeah.
now that sounds like a band you'd hear on late night space radio or something.
Haha, right.
Yeah.
Catchy name, but yeah, it's a little more complicated than that.
I figured. So EMT, what does that even stand for?
It stands for epithelial to mesenchymal transition.
Epithelial to mesenchymal. Okay. Gotta love those science terms.
Right. But it basically boils down to cells changing their identity, their whole behavior, really.
Okay, so it's like not just different neighborhoods of cells anymore, but cells that can actually, like transform themselves?
Yeah, kind of like a caterpillar transforming into a butterfly, you know? They can make themselves more aggressive, more invasive.
Oh, that doesn't sound good. So those quiet little cells hanging out, minding their own business, they can suddenly go rogue, start spreading around the body.
Exactly. That's why understanding these RHPs is so important.
Yeah, way beyond my pay grade. Okay, so we've got these shape-shifting cells running around, and there's 3 different types of them, right? The EMT trio. So what makes each one unique? What are their, like, superpowers?
Well, one of them, they found it mostly in melanoma.
The melanoma, okay.
And this one was linked to drug resistance.
So these melanoma cells, they're like, what, evolving in real time to dodge the treatments we're throwing at them?
Yeah, you could say that. That's why just knowing what kind of cancer someone has isn't enough anymore. We've got to understand the specific programs running the show in those cells.
It's like, okay, we know you're dealing with a criminal organization, but knowing their tactics, their methods, that's how you take them down. So that's one member of the EMT trio. What about the other 2? What kind of trouble are they causing?
Well, another one, they found it mainly in head and neck cancer.
Head neck cancer, okay. And this one is all about
This one was all about metastasis.
metastasis, right? So that's when the cancer cells break away from the main tumor and spread to other parts of the body.
Exactly. It's a big reason why cancer can be so difficult to treat.
So this RHP, this is like the getaway driver for the cancer cells.
Yeah.
Helps them escape and set up shop somewhere new.
Aha, that's a good way to put it.
Okay, so we've got the drug-resistant one and the getaway driver.
Right.
What's the last member of the trio up to?
This one's the most mysterious of the 3. They found it in cells that weren't dividing.
Wait, really? So not growing, but not just chilling out either. What are they doing?
That's the million-dollar question. They were active in some way, but it wasn't clear exactly what they were doing.
Oh, that's unsettling. They're like sleeper agents, just waiting for the right time.
Yeah.
Okay, so we've got stress response, shape-shifting cells. What else did this research unearth?
Well, we should definitely talk about the senescence surprise.
The senescent surprise. Okay, you've got my attention. What's so surprising about it?
It's about these cells that have basically entered retirement. They've stopped dividing.
They're putting their feet up, taking it easy. Sounds harmless enough.
You'd think so, right? But here's the surprise. These retired cells, they're not just sitting around, they're actually pumping out all sorts of signals that can influence the other cells around them.
Hold on.
Yeah.
Even though they're not dividing anymore, they're still messing with the other cell.
You got it. It's like they've retired from active duty, but now they're acting as advisors, still pulling the strings behind the scenes.
Wow, that's kind of creepy. So even though these cells aren't dividing, they're still part of the problem. Maybe even driving tumor growth in some roundabout way.
It really highlights how interconnected everything is within the tumor. And speaking of complex, there's one RHP that really stood out because it could have a big impact on treatment. The Epithelial Senescence Associated Program, EPISEN for short.
EPISEN, okay. So is this another one that has to do with those retired cells? The ones that have stopped dividing? What makes this one so special?
Well, what's interesting about EPISEN is that the research suggests it could help us figure out which head and neck cancer patients would respond best to a certain type of therapy. Cetuximab, it's called.
So we could know ahead of time whether or not a treatment is likely to work before we even start. That's amazing. No more just throwing things at the wall and seeing what sticks.
That's the goal. This is what we're talking about when we talk about personalized medicine, tailoring treatments to each individual patient.
So how does this EPISEN thing work? How does it actually affect how well a treatment like cetuximab works?
Well, it all comes back to those signals that the senescent cells are sending out, remember?
Right, the senescent cells are still gossiping with the other cells, even though they're not dividing anymore.
Exactly.
So what are they saying? How does that affect how well this cetuximab treatment works.
It's like they're speaking a whole different language, a chemical language.
Yeah.
And the epicene cells, they're especially chatty. They send out this whole cocktail of signals.
Okay, and what do those signals tell the other cells? Like, what's the message?
Well, it's not always a friendly message. Sometimes those signals actually tell the other cancer cells to grow faster.
Wait, really?
Yeah, they can even tell them to become more invasive or even hide from treatments.
So even though those epicene cells are kind of retired, they could actually be making the tumor more aggressive, more resistant to treatments.
It's definitely a possibility. It shows just how complex this whole tumor microenvironment really is. But here's where it gets really interesting with cetuximab.
Okay, I'm listening.
So cetuximab, it targets a specific protein that's involved in cell growth. And the research found that when epicen is present, the tumor might actually be more sensitive to having that protein blocked.
So it's like the epicenter is actually helping the cetuximab do its job.
That's a good way to put it. It's kind of counterintuitive, but those signals that the epicene cells are sending out, they might actually be making the tumor more vulnerable to this particular treatment.
Wow, that's wild. So instead of just trying to kill cancer cells, we could actually target these programs, these RHPs, and maybe even use them to our advantage.
Now you're getting it. It's still early days, but this research opens up so many possibilities.
It's like we're not just playing checkers anymore. We're playing 3D chess.
Right.
We're learning how to manipulate the whole board, not just take out individual pieces. This is mind-blowing stuff.
It really is, and it all comes back to understanding the complexity of cancer.
Yeah, it's not just one disease. It's a whole bunch of different diseases, all with their own quirks and challenges. But we're finally starting to crack the code one cell at a time. This research, it's like we've just discovered a whole new continent. We're just starting to explore it. But what we find there could change everything.
I couldn't have said it better myself. It's a brave new world of cancer research.
Well, there you have it, folks. Another deep dive into the cutting edge of science. We hope you found it as fascinating and mind-blowing as we did. Until next time, keep those brains buzzing.